I put some links in the chat, the artifacts related to this work. So what we do at Dotphoton is trying to kind of merge the physics of optical imaging with machine learning. And this particular work stream is around the integration of kind of the workhorse that we have in machine learning, which is gradient or backpropagation, with the hardware and the physical models that we have in optical imaging. And what I would like to do today is introduce a few concepts that we discovered and kind of digested along the way at this intersection, and also show a path that we are taking towards making hardware cheaper through better software when we optimize the acquisition for machines in particular, rather than humans. There's a paper that I put in the chat for like full detailed account of the experiments and the work we did here. And I will give a quick rundown of the main concepts. So my background is in machine learning. So for me, working together with physicists and people in microscopy is always like a mutual learning experience. And just to give a bit of a perspective, how we came together is what you typically have in machine learning is the part here on the right, a machine learning model. You do some standardization, some augmentation, but we add the component here from the hardware itself, which is what we call the data model. So people usually don't have access to this. In machine learning, they start with some version of the processed image and then do augmentations on that to get whatever they want downstream, their classification score, like you see here. But what we want to open up is all the things that happen on hardware. So that relates to the measurement and then the ISP, which goes from the raw sensor all the way to the processed output. And in particular, this middle part, there is a host of variations. Sometimes this is done on the device. Sometimes this is done in software later. And it introduces a number of challenges and opportunities as well when it comes to machine learning integration. The challenge mainly is around variations in the image statistics. So as you probably know, machine learning models, they have a very high capacity to find patterns, but they also easily latch onto spurious features, which is actually just what they are intended to do. They try to extract patterns that help to solve the problem. They don't really care about causal or underlying biological relationships because they only look at image statistics. You can regularize against that a bit, and there's some tricks to kind of circumvent these problems. But in general, as long as we don't have this detailed technical knowledge that usually is present on the hardware side in microscopy, it's really hard to test models for that or also prevent them from becoming sensitive, right? So if we have one microscope in a hospital A and another microscope, or even with same microscope with other settings in another hospital or lab, it can be very challenging to use the same machine learning model. So that's the kind of the motivation on the challenge side. On the opportunity side, what we are interested in is to find ways in which we can improve the data. So you could call this data optimization to help with the downstream tasks. So a lot of the microscopy processing is, as I said in the beginning, optimized for human consumption. But if we assume that there are certain workflows in automating the analysis of the data, then we can look for data representations that are good for the machine learning model. And that's the kind of the scope of this whole work. And I will quickly go into each of these applications to showcase the results, what we learned, what worked, what didn't work, and what you can do with the existing code, and what we are currently working on the research side in terms of future paths. So let me maybe quickly preface this, and because it is often there's a lot of talk about distribution shift, data drift in machine learning, and it usually has a probabilistic notation or perspective where we just assume we have some training data following a distribution P, and then in practice we might get samples that are drawn from a different distribution Q. Where this view is falling short is in actually characterizing what the differences are, and this is non-trivial. And here the physicists and microscopy community has a huge advantage because they have very intimate knowledge of the data model, and that's also how we explicitly characterize drift in this work, is that we assume a different processing model underlying the output data that goes into the machine learning model, and that's the drift component. So if we specify what you see in the figure here, in this middle part, if we specify that explicitly, we can have very fine-grained control over variations in the data, because we can account for all these different factors and have it explicit. And with this in mind, we conducted a bunch of proof of concept experiments here on some blood smears with bright field microscopy. And the idea for this drift synthesis application is really what I just kind of outlined in terms of use case. You have some raw reference data and you want to emulate different versions of that data as if it was captured under other or processed under other ISPs. And what this gives you then is from like one reference version, you can just on the fly create physically or metrologically accurate variations of the data and do that testing to your heart's content. And this is the type of result or the way this result is then presented. So you, on the one axis, have all the different data models, all the different processing variations of the reference raw measurements. Then with the downstream machine learning task model here, a classifier that detects cells trained on that data model data and tested against the same variations. And this allows you to do model selection. So imagine, like I said, you have a bunch of labs or hospitals across which you want to deploy a machine learning algorithm. You would want to beforehand test the model against all those different acquisition settings. This is something that is captured with this workflow. Similarly, you might want to optimize a data model for a certain deployment setting, and this you can also tolerance under this drift synthesis with the different configurations that are displayed. I just want to give a quick, maybe quick remark also on the figure on the other side. For those people not so familiar with machine learning and the kind of culture and literature around robustness. So what is very common to do in machine learning is to just apply additive noise to the processed data. So for here. So instead of accounting for an accurate data model, what people often do is there is a catalog of additive noise that people like to kind of use as a benchmark, and then they have some processed data and they train their model, and then they throw all kinds of noise on their test data to estimate the effect, the robustness of the downstream model to that noise. The problem is obviously that this is not equivalent to, for example, even injecting a Gaussian additive noise at either one of these steps, because there are nonlinear transformations along this pipeline, and so it's not equivalent to inject it at an intermediate step versus at the end of the pipeline. And that's the kind of, for the machine learning world, the comparison we draw here is that you have to be very careful and metrologically accurate to kind of get to the right conclusions in terms of model selection, because if you use the wrong noise model and just some arbitrary augmentations that don't reflect reality, you will pick the wrong acquisition model, you will pick the wrong machine learning model. So it is very important to try to leverage the knowledge that exists in hardware and in physics for these types of tests. So that's the first major use case. And the second one is related, but there it already gets a bit more interesting, because we can actually leverage the gradient connection. So again, maybe just as a little disclaimer, what is the gradient? You take the derivative of a function with respect to some parameter or the input, and that's how you typically train this part here. When making this whole thing differentiable, the nice thing is that we can do what we called here drift forensics, which is basically just an adversarial search on the parameter space of each of these processing steps. So what does this mean? Imagine this function here, the color correction, or the sharpening has some free parameters that you can set, and making them differentiable means that we can train our model like usual, this thing here, and once we're done, we can take new test data, pipe it through this entire pipeline, and then search on the parameter space of any particular step in this processing. So let's take the sharpening, and find a parameter configuration under which the performance of the downstream task model breaks. And that allows us to kind of build these types of forensic signatures that you see here at the bottom. So we do this forensic search for each step in the processing pipeline on the parameters, plus all we do it individually, but also on all the parameters combined. And then you get this type of a profile for the forensic result, where you can see that for some settings here, the downstream task model is very sensitive to parameter changes, whereas for other it is not. And this you could then give to a prospective user to say, look, be careful when you vary the Gaussian blur during acquisition in your ISP, because we know that the model is somewhat sensitive to this. For the others, you don't have to be so concerned. Or you can use this knowledge and go back and say, okay, there we have some sensitivity, let's retrain and try to make the model more robust to that. So it gives you like a very model-specific profile of the sensitivity to the processing that you apply during acquisition. All right, and so now I will try to kind of give a short overview of the last one, but I also see we are running out of time.
Yeah. If you could wrap up, please. Thank you.
Yes, so we can move on. But the idea is very simple. It is the same thing basically, but we don't just push the gradient into the data model for the analysis of the sensitivity, but we actually optimize the data model itself. So we make the data fit the downstream task model and do a joint optimization. And as you see here for the microscopy use case, this has a very big performance gain and also a more stable learning trajectory. So this is the blue line here, the learned setting versus just frozen, like a normal static ISP. But I also want to point you to some caveats here. So we have like another kind of benchmarking task with some drone data, which is just segmentation, like a regression problem for cars. And here we don't see any performance gains at all. So it's very task dependent. And I think that's an important takeaway for that one. All right, so not to take away any more oxygen, this is kind of the quick rundown. If you are interested, like we are also quite active in research. We are building together with other colleagues, like a big community around this data-centric machine learning, where we host workshops and things like that. So yeah, if you are interested to contribute to that, please reach out or just visit the website. And yeah, it's open, it's research focused, and yeah, that's it. I will try to, if there's questions, maybe do it in the chat so that we can move on to the next thing. Thank you so much.